Hi, my name is Giordan. I'm a graduate student in the lab of Dr. Fidnat Yildiz at the University of California, Santa Cruz in the microbiology and environmental toxicology department. In the Yildiz lab, we study the bacterium Vibrio Cholerae, which is the causative agent of the acute diarrheal-disease cholera. Cholera can result in hypotonic shock and death within 12 hours of the first symptoms. Ingestion of Vibrio Cholerae in contaminated food or drinking water is responsible for up to 4 million cases of severe diarrhea and up to 143,000 deaths every year. Cholera occurs in areas where access to clean drinking water is limited. Many areas in the world still lack access to clean drinking water and sanitation today, making cholera an ongoing problem worldwide. Vibrio Cholerae is a facultative human pathogen, transiting between the aquatic environment and the human host. An important process the pathogen utilizes throughout the infection cycle is biofilm formation. Biofilms are surface-attached microbial communities encased in a self-produced extracellular polymeric matrix. Biofilms are preferred mode of life for many microorganisms, as growth in a biofilm confers many benefits compared to growth as free-living planktonic cells. The ability to form biofilms enhances environmental persistence, transmission, and infectivity of Vibrio Cholerae. Cells grown in a biofilm show increased abundance in the intestine during host colonization compared to those not grown in a biofilm. This is shown here during visualization of the intestine, shown in blue, with Vibrio Cholerae cells shown in yellow. Now, biofilm formation in Vibrio Cholerae is a complex process, and so it must be tightly regulated. One tool used for this are two-component regulatory systems, or TCS. Now, TCS allow bacteria to sense and respond to changes in the environment. In response to the presence of a relevant input signal, a sensing protein is activated, which then activates a response regulator protein, which can go about eliciting an appropriate cellular response. Now, so a goal in the Yildiz lab is to identify and characterize TCS contributions to biofilm formation. And my current research focuses on one of these systems, known as RvvAB, for regulator of VPS and virulence. This system was identified in our lab during a screen for increased biofilm gene expression when individual sensing proteins were absent. The absence of the sensing protein, RvvA, shown here in blue, led to a 14-fold increase in expression of the biofilm gene VPSL compared to the wild-type strain shown in black. RvvA's impact on biofilm formation was clearly visualizable with confocal microscopy, where biofilm formation with RvvA absent led to increased roughness and microcolony formation, which is associated with increased biofilm production. Further work has found that RvvA also positively regulates virulence in Vibrio Cholerae. Its absence led to a defect in host colonization, and some additional data from our lab suggests that this system may be involved in utilization of a host-obtained nutrient source. Moving forward, I hope to further characterize this system, its activating signals, and its mechanism of action, therefore furthering the understanding of this important human pathogen and how to combat it. And with that, I'd like to thank ARCS and the entire Yildiz lab for making this work possible, and thank you all for listening.